Welcome life science learners to another lesson. Trust that you guys are well and looking forward to our lesson today. In our lesson today, we're going to be focusing on revising a very important topic, DNA and the code of life. It's important that this concept is reviewed constantly. It often forms the basis for many other topics to come. So let's get straight into the revision lesson. Let's have an overview of the sections and then we'll apply some of our understanding. It's important that you, as you sit at home, are able to make notes. I often remind you to have a set of terms that are important. And so that's where we start in our lesson today. We're going to go through a list of terms, help us to refresh all of these terms so that when we get into applying these terms in questions, it becomes easier. So let's overview some terms and then we'll apply our knowledge in answering some questions. So it's important that we understand the overview of DNA. We're going to spend some time looking at the processes of protein synthesis and replication, and then we'll get on to discussing mutations, and also then spend some time looking at some revision activities. As I mentioned, terminology is fundamental to performing well in life sciences. It's important that you draw up a list of these terms. As I have here, I've indicated some of the most important terms that frequently appear in this topic. So let's get through them, revise, recap, and I hope this tickles those memory cells so that these terms come back. So let's get into that. So guys, the concept of chromatin network, again, go back to the nucleus, and that's where we started off in the year when we spoke about DNA being the molecule of life, and essentially that's in the nucleus where all the chromatin network is found. We obviously spoke about chromosomes and how those chromosomes or the DNA arranges itself into these compact bodies called chromosomes, which are essentially made up of proteins, histone proteins, around which the DNA is wrapped. So we do have the histone proteins as a construction protein of the chromosome, and we find that the DNA is wrapped around that to form these complex structures called chromosomes. In this specific chromosomes, we mentioned the term chromatids, and essentially chromatids refer to those copies of DNA that have been produced after replication and then held together by the centromere. So it's important that you recollect the structure of the chromosome, that it's made up of chromatids and the centromere that holds the chromatids together. And that's important in the next section when you look at meiosis and the revision around that. The concept of a gene is very important because often that relates to genes and how we understand the concept of inheritance. So a gene is again a sequence of nucleotides that code for a characteristic, and we see the importance of that in terms of protein synthesis and how proteins are made. It's important that we understand what a gene is and what happens to the gene when a mutation takes place. So it's important to recognize what a gene is. A nucleotide again refers to the basic building blocks of nucleic acids. And remember, the two types of nucleic acids are DNA and RNA, and so we talk of them being made up of these simple building blocks called nucleotides. The nucleotides contain nitrogenous bases, and we refer to them as your ATGC base pairs, and so that's important when you're looking at adenine, thymine, guanine, and cytosine as being your DNA bases or your nitrogenous bases, as well as adenine, uracil, guanine, and cytosine for your RNA nucleotide bases. So that's a very quick recollection of the nitrogenous bases in both DNA and RNA. Again, a recap of what DNA is. You should know the structure of DNA. You should know the similarities between these two molecules in terms of the location, where they are, what are they made up of, and essentially the differences between them. And often we tend to revise that section towards the end where we talk about the differences between RNA and DNA, often in a table, and that's an important comparison to have. The significance of mitochondrial DNA in the mitochondria and its link to maternal ancestry, and that's important, and how we use that to determine relatedness along the maternal line, as well as we talk about the different types of RNA, where we've mentioned messenger RNA, transfer RNA, as well as ribosomal RNA, which we see all being important in the process of protein synthesis. Again, a quick overview is that the ribosomal RNA is our RNA, which is what the ribosome is, and that is an important part when we discuss protein synthesis. We mentioned the term complementary base pairing, and that again refers to how the bases connect on a double-stranded DNA. And again, the terms purine and pyrimidine need to come back because those are important in terms of identifying the different types of bases. And a very important concept, DNA replication, as an important process that we need to understand, as well as chromosome mapping in terms of the Human Genome Project. Okay, let's have a very quick overview of the process of DNA replication. It's important that we remember that DNA replication occurs in interphase, and it's a process in which DNA undergoes copying. And so it's important to remember these keywords where DNA unwinds and unzips, exposing the DNA templates. And these templates then become available so that new or free DNA nucleotides can base pair with these templates. We also recognize that the base pairing is facilitated with an enzyme, and that enzyme allows for base pairing to occur in a complementary nature, and we discuss all of that, and eventually we talk about how the base pairs link up together and how the DNA helix reforms. And this often comes in a question for about 4 to 7 marks where you describe the process of DNA replication. So you need to know what do you need, when it takes place, and how it takes place. So again, the enzymes, the free nucleotides, as well as the DNA templates, and it happens during interphase, and that's essentially the importance of that process, looking into mitosis and meiosis. It's important that the next concept is protein synthesis, and you review that concept well. And the best way to revise this section is actually to have a few diagrams that illustrate the two processes that are important in protein synthesis. And we'll have a quick overview of that: transcription and translation. And a reminder that transcript, again, is the formation of a message, and that is mRNA. And translation is changing that mRNA into a sequence of polypeptides. And we refer to those sequence of polypeptides being a chain of polypeptides, which will eventually combine with other polypeptides to make what we refer to as proteins. So that process, we'll very quickly overview that. You should know that there's a difference between where transcription happens, which happens in the nucleus, and the process of translation, which happens in the cytoplasm. What's important is that we recognize that transcription is when a gene produces a copy of itself which codes for information, and that copy is mRNA, and that mRNA then leaves the nucleus to move to the ribosome, where that ribosome is going to process the mRNA and read each codon and bring in specific tRNAs with their unique amino acids attached to them. The concept of codon and anticodon base pairing is important, and then being able to understand how amino acids are put together in that process. So guys, that's a very quick overview and a recap of some of the important concepts. Again, if you do not recollect these concepts, go and prepare some diagrams, look at your textbooks, and try and write down a few points around these 3 major processes: DNA replication, protein synthesis, where we talk about transcription, as well as translation. And so that's an overview. Let's have a short break, and then when we get back, we will apply some of this understanding to a few questions. Stretch around, get some water, and I'll see you in a bit. Welcome back, life science learners. Trust that you've been able to refresh yourselves. Let's get back into our lesson. Again, we're spending some time revising the basic concepts around DNA as a molecule and its importance as a code of life. We've discussed some important terms and concepts such as DNA replication as an overview. We've looked at protein synthesis as an overview. As I mentioned, it's important that you prepare some notes on these and understand how these processes occur. They're very important in terms of preparing for a question on an essay where you need to describe DNA replication, or for that matter, protein synthesis. However, in this segment, we're going to spend some time looking at how we apply our understanding around the structures of DNA and some concepts around how DNA replication occurs. So let's get straight into it. I encourage you to pull out a book, and if you can write down some answers as we work through them, I'm sure we can check these as we get through the lesson. So the first question, if we look at it, the diagram alongside represents a portion of a DNA molecule. So it tells us that it's a portion of a DNA molecule. If we look at it, we can clearly see that it's got two strands on either end, and there's some base pairing that's occurring right here in the middle. So we do know that it is DNA as it's mentioned in the question, so we don't have to figure out whether it's a DNA undergoing process called what we refer to later on as protein synthesis. So this is just a simple DNA molecule where we see it's double-stranded. It's important that we recognize the different labels. And I often encourage the learners when they see a diagram to try and understand what are the different parts before getting into the question. So I can clearly see that there's an area that's circled having three different parts. We can see a structure here that's labeled B. We can see a structure labeled C and D. So let's get straight into some of the questions based on this. So as anticipated, identify B and C. So guys, if we look at structure B and if we look at structure C, we can clearly see that it is pointing specifically to what we refer to as B points to the complementary base pairs. Now, obviously this is a DNA molecule, so that's going to be a DNA nucleotide that it's pointed to specifically. When we look at structure C, we got to recollect that DNA is double-stranded and it's made up of a sugar phosphate backbone. So here we find the sugar molecules, and here we find the phosphate molecules that form the sugar phosphate backbone. So we know that in one single nucleotide you have a nitrogenous base, we have a sugar molecule, in this case it's deoxyribose sugar, and we have a phosphate molecule here. So molecule C points to a phosphate molecule, and molecule, as I mentioned, B in that previous diagram pointed to a nitrogenous base. Okay, let's continue with this question. A, 1.2, monomer A. Now guys, the term monomer is another word for the concept of building blocks. Now remember that DNA is made up of building blocks, which we refer to as DNA nucleotides. And those nucleotides consist of three different parts, as I've mentioned in this illustration. It contains a sugar, it contains a phosphate, and a nitrogenous base. So the building blocks, or another word for building blocks, is monomer. In this diagram, monomer A is a DNA nucleotide. And I've got to be specific because this is a DNA molecule. Often we get learners simply replying, it's a nucleotide. And I think it's important that at this point that you're quite specific in being able to differentiate that it is a DNA nucleotide and not just a nucleotide, because it could represent an RNA as well. So the correct answer would be a DNA nucleotide. And it's important that you are able to express yourself correctly and scientifically right. Let's move on to B. Two scientists who received a Nobel Prize for the discovering of the DNA molecule. Guys, it's important that we recognize that DNA was discovered collectively over a period of time in different stages. However, there were three significant scientists that contributed to the discovery, and we go back into the history of that. We know that Watson and Crick, so the fellowship between James Watson and Francis Crick, were recognized for that, obviously, when they put together the model of the DNA. We also know that Rosalind Franklin was posthumously recognized for her contribution to the discovery of the DNA. So there were three significant scientists that played an important role: James Watson, Francis Crick, as well as Rosalind Franklin, in terms of her process of being able to crystallize DNA and photograph that. So again, you've got to be able to identify this. It's important that we recognize scientists in their endeavors and we recognize them in terms of their contributions to science. The next question: one organelle in a cell where DNA is located. Guys, it's important that when we revise DNA, we recognize that DNA is found in different structures. For example, we've been looking at the nucleus. However, we do know that DNA is also found in the mitochondria, and it plays an important role in how some of the genes in the mitochondria are expressed. We also know that plant cells in their chloroplast contain some DNA, and that DNA plays an important role in the expression of those genes, or those producing important characteristics in those specific organs, or organelles rather. So let's recognize the importance of these. So we mentioned nitrogenous bases as being structure A. We also mentioned that these individuals, James Watson, Francis Crick, Maurice Wilkins, as well as Rosalind Franklin, much later was recognized for that. The organelles in which the DNA can be found included the nucleus, the mitochondria, and in plant cells, the chloroplast. Let's move on to question 1.3. Describe how a mutation on DNA may change the structure of a protein. And that's a 4-mark question. Guys, it's important that we recognize that mutations, again, the definition of a mutation is a change in the sequence of a nucleotide, of a gene. And that affects a single nucleotide, or it could affect more than one nucleotide. So DNA is often affected by mutations which the cell generally corrects. However, sometimes these mutations go by uncorrected, and that we refer to as an alteration in the sequence of a gene. So let's try and unpack how you would answer this question. We've got to recognize that genes code for the sequence of amino acids that make up a protein. So when there is a change in the gene, it's going to in turn affect the chain of amino acids. Well, it may affect, depends on the type of mutation that takes place. So let's try and unpack how you would answer a question like this. It's important that we recognize that, I'm going to change my pen so it makes it easier for us to differentiate, that DNA will code for characteristics. So when the DNA changes, and that happens because of an alteration or a mutagen, we say that a mutation has taken place. This leads to a different mRNA being produced, or a change in the codon. So we do know that when DNA is being copied into what we refer to as mRNA, if there is a mistake or a problem in the process of it being copied, and that's mRNA, if there's an alteration in that, or if there's a mistake that happens at that specific point, it in turn, when it leaves the nucleus, carries what we refer to as an abnormal or a problem in this. So that could be an altered nucleotide on the mRNA. And this means that this entire codon has now either changed or could be coding for the same amino acid. So we refer to that as a different mRNA being produced, having a different code. Okay, this will match with a different tRNA and produce an anticodon that does not carry the initial amino acid that was coded for by the unaltered codon. This results in a different amino acid being brought by the tRNA to the ribosome. In turn, this leads to a different protein or an altered protein. So guys, we do know that mutations can be neutral, where a mutation like this sometimes codes for the same amino acid, and we refer to those as neutral mutations. We also know that certain mutations can be harmful, where it affects, in this case, an important protein. And we know that mutations like sickle cell anemia, which affects the shape of the red blood cell, is a harmful mutation. We also know that sometimes certain mutations can be beneficial, where it creates a protein that gives some individuals an added feature. For example, there are some populations in the world that have a gene that is mutated and made them resistant to HIV. So again, that's a beneficial mutation if one has to look at the benefits of being resistant to HIV. So again, we need to recognize that mutations have both neutral as well as harmful or beneficial effects. And that's essentially an answer to that question. Okay, the next part would be tabulate two structural differences between a monomer of RNA and a monomer of DNA. Guys, the key words are structural differences. So it's important that when we compare the differences is that we talk about the structure. We cannot talk about the location of where the DNA is found in RNA, because that's about the location. Let's reference the structure in that specific answer. So you could answer by describing that mRNA contains a ribose sugar, whereas DNA contains a deoxyribose sugar. We also know that in mRNA you have adenine, uracil, guanine, and cytosine. So we talk about AUGC, whereas in DNA you have your adenine, thymine, guanine, and your cytosine, where thymine and uracil are the two bases that are different. Okay, so let's move on to the next question and let's apply some of our understanding further on to this process of protein synthesis. So the diagram below represents a process that occurs during protein synthesis. Now guys, remember that as I mentioned, protein synthesis happens in two parts. The first part is what we refer to as transcription, where in the nucleus an mRNA is produced. A transcript of the gene is produced as mRNA. That then moves into the cytoplasm, where we know the process of translation occurs, which is putting together a chain of amino acids with information that comes from the mRNA. So here we're looking at this illustration that shows you these ribosomes, and these ribosomes are often made up of 2 units, and I often like to use the word the snowman, and if you remember that, you can easily identify what we call a ribosome. So here we're seeing a ribosome, 2 of them actually reading through a strand of mRNA which has these codons on them. We also can see that there's some tRNA molecules onto which unique or specific, I'd like to refer to them as amino acids attached, which you can see here. And these are based on the codon and anticodon base pairing. So that's an overview of this illustration. And let's see some questions on this. So the question is, identify the process above. And if we look at the process above, guys, as I mentioned, this process is certainly happening at the ribosomes outside in the cytoplasm. So that process again refers to the process of translation, where we see a chain of amino acids being produced, and that again is a key part of the final product of translation, where we find the amino acid sequence put together. Okay, let's look at the next question. Identify organelle A. So if we look at organelle A, as I mentioned, guys, it's a structure that is important in protein synthesis, and we refer to that as the ribosome. And I often remind the learners to think of a structure that's made up of 2 units. It's got a small subunit and a larger subunit, which is often a unique feature of these ribosomes. They're 3-dimensional structures, guys, in which the mRNA moves through. And that's essentially what is being illustrated in this diagram. The next structure we need to identify is molecule B. And as I mentioned, that during the process of transcription, we find that mRNA is produced, and that mRNA leaves the nucleus and moves into the cytoplasm, where the ribosomes read that. So clearly we can see that the ribosome is processing this mRNA molecule. Several of them read this mRNA molecule and then bring in a specific tRNA with its amino acid to it. The next question is bond E. Okay, so we know that amino acids put together form a polypeptide, meaning a chain of amino acids. Often we ask, what are the bonds that hold these building blocks together? And learners often tend to confuse the term polypeptide chain and polypeptide bonds. Unfortunately, you've got to mention that it is a peptide bond that is found between your amino acids and not a polypeptide bond. That's the polypeptide refers to this chain of amino acids that make up the final sequence of amino acids that form in that protein. Okay, so as I mentioned, that's a quick check of our answers. And as we wrap this up, let's very quickly look at one more question. Give only the letter of the molecules that carries the amino acids. So letters that carry the amino acids, if we look at it, this would be a structure that's carrying the amino acids, so it would be letter C, and that structure is referred to as the transfer RNA. It's copied from DNA. We know that mRNA is copied from DNA, as I mentioned earlier on, so that's going to be structure B. And finally, is this monomer or building block of protein? So is the monomer or building block of protein? And so we know that these are the monomers or building blocks of proteins, and that would be structure labeled D. Welcome back, life science learners. We're busy with revising DNA, the code of life. Let's continue with our application of our understanding by attempting some questions. It's important that we now look at revision activities and test some of these important concepts that we've been revising. Let's look at this question. The diagram below shows the process of protein synthesis. As I mentioned, it's important that we read the question clearly, and that often points to processes that are seen. So this diagram illustrates different processes, and they're labeled here process V and process W. And it shows you different structures. And as an overview, I'd like to step back, look at this, and try and see structures that I can easily identify. What I notice is that it points to a membrane, and I can see that it often is the nuclear membrane with these nuclear pores. And that's a giveaway for the structure in terms of where process V is occurring. And outside process V is this area, which I often recognize as the cytoplasm. So we clearly see two processes occurring, one inside the nucleus, which is clearly indicated here, so that's inside the nucleus, and then the second process outside in the cytoplasm. We've seen several structures that are familiar now. We know that, as I've mentioned, we can recognize this structure as a ribosome. It's good that we're able to recognize that early on in the question. We know that these structures, which often are T-shaped, play an important role in bringing in these molecules here, which are the building blocks of proteins, which are amino acids. So let's get into the question and see what has been asked. So the diagram alongside shows the process of protein synthesis. Question 1. Identify the following: molecule X and organelle Y. And that molecule X, if we go up, guys, if we look at it, you can see that it's double-stranded. It's a complex structure having two strands. And for me, I know that the only structure in the nucleus that is double-stranded and helical is the DNA molecule. So structure X is the DNA molecule that we can easily identify. If you asked for reasons, then I'll talk about it's double-stranded. If you can see evidence around the base pairing, and if you can see that there's adenine and thymine, that's another reason that you can confirm that it is DNA. The next part with organelle Y. So guys, it's important that we remember the term organelle. Again, we don't often use the term organelle, but in the cell, we know that there are different parts that perform specific functions. And so this question points to organelle Y. And if we look at organelle Y, guys, as I mentioned earlier on, it's important that we recognize that this organelle contains two subunits, a small subunit and a large subunit. And that is typically an indication of an organelle called the ribosome. So that would be the ribosome. So as I mentioned, X was the DNA and Y was the ribosome. If we look at question 3.2, identify the nitrogenous base labeled 1 and 3. Now guys, so we've got to go onto the diagram, and you can clearly see here is nitrogenous base 1. Let's try and remember exactly what structure that is. So that is a tRNA molecule that has the nitrogenous bases on it. It's important that we first identify the type of nucleic acids, because we do not want to confuse the DNA nitrogenous bases with the RNA nitrogenous bases. So we've clearly identified that structure on which number 1 is a tRNA molecule, and hence we've got to remember that it could either contain adenine, uracil, guanine, or cytosine. Now, in order for us to be able to determine what that number 1 is we recognize that on the tRNA, we have what we refer to as the anticodon. And so that anticodon is complementary to a codon that we see on the mRNA. So in order for us to be able to identify structure 1, or the base pair 1, we've got to look at this complementary base pairing. And as we look at that, we can clearly see that it has C on the mRNA, and we've got to figure out that. And it's important, as I mentioned, in the complementary base pairing between DNA and RNA, or between the mRNAs, the codon and anticodon, that we know that guanine base pairs with cytosine. Okay, so that number 1 would be guanine. If we look at number 3, here on number 3 we've got an adenine, if I read that correctly. So it means that we now have to put in a uracil and not thymine, because we don't have thymine in mRNA or in the tRNA molecule. So that's going to be uracil. And it's important that you recognize the difference between the two. Describe the role of DNA during transcription. And that's a 3-mark question. Guys, it's important that we recognize that DNA codes for life. And DNA codes for genes, has genes that code for specific proteins. And essentially, that is what the question points to. So let's see how you would answer that question. So it's quite an elaborate explanation, but it's important that you're thorough and distinct about how you explain the process. So as I mentioned, DNA codes for characteristics, or DNA has genes. We know that the double-stranded DNA unwinds and unzips, those important concepts of unwinding and unzipping. What we need to recognize is that when that unzipping occurs, that the hydrogen bonds break and exposing the strands, or now forming the new templates. And we see that one strand is used as a template to form mRNA. Using the free RNA nucleotides, so those are the ones that are floating around in the nucleoplasm, the enzymes will base pair with their complementary DNA nucleotides on the template. Eventually you find that the coded message is then sent to the ribosome in the form of mRNA. And that is how mRNA codes for protein synthesis. So guys, that's an overview of this section where we've looked at protein synthesis and how it takes place. Let's get into a short break, and then when we get back, we'll look at some detailed processes like DNA fingerprinting and how to attempt to answer an essay on this topic. So quick break, and I'll see you in a bit. Welcome back, life science learners. Trust that you've refreshed and ready for this part of the lesson. It's important that now we look at some of the applications of understanding how DNA works. We're going to attempt to look at DNA fingerprinting. We're also going to spend some time looking at how to potentially answer a question based on DNA. It's important that we now take all of those concepts and put them together and link them to various processes such as DNA fingerprinting, writing an essay, and the applications of that to processes where mutations take place. So let's get straight into the lesson and see how this part of the segment unpacks. So the question reads, and I often remind the learners to highlight key components of the question. And we'll go through this together. Detectives were investigating a crime scene and they found blood on a broken window. They suspected that the blood was that of a criminal. To identify the criminal, they analyzed a DNA sample from the blood and compared it to that of 4 suspects. The diagram produced is on the next slide. So guys, here we're seeing what we refer to as a DNA fingerprint, and here we're seeing the sample of DNA that was extracted at the scene, and we're seeing 4 possible suspects that have been analyzed, and their samples have been tested, or what we call DNA fingerprinted. And it's important that we recognize that DNA fingerprinting is a technique that has revolutionized how we can identify individuals, link them to parents, look at missing identities of individuals that have been found, deceased individuals. Also in the conservation of animals, where trade of animals such as the horn, rhino horns, we can now use that to link that to specific organisms or animals that have been poached by poachers. So it's been an amazing technique, and now let's look at how we answer a question on that. So the first question on this, name the technique that was used to identify the criminal. So guys, this technique is called the DNA fingerprint or fingerprinting, and it's essentially it's a process that we use to identify individuals. We use the rationale that no 2 individuals have the same genetic makeup, and hence, when DNA fingerprinting takes place, we know that it will produce a unique band based on an individual's genetic makeup. if we see that two banding patterns, and I refer to these as banding patterns, which are produced by segments of DNA that have traveled through a gel that is being created, and that separates out the DNA based on their sizes. And so that separation creates what we call a DNA banding pattern. And that banding pattern will be unique to each and every individual on Earth. And that is the uniqueness that we use in differentiating individuals. Okay, so the technique is called DNA fingerprinting. Who is the possible criminal in this specific DNA fingerprinting band? So guys, the best way for us to be able to link an individual to a crime scene is to have evidence. We know that DNA can be used as evidence to link an individual to a crime scene. However, it does not conclusively prove that that individual committed the crime. So part of convicting a criminal is being able to find evidence that links him to the crime. DNA is one part of that evidence. So here we've got the sample and we've got 4 different suspects. And so I'm going to go through this and I'm going to compare each band, and that's essentially what I'd encourage you to do, is to use a ruler and place that along and let's see. So as we look at this band, we identified that these two are together. So it's definitely not, there's nothing there, there's something here. There's certainly an identical match between that, and definitely Ntabi, who's got only one, is not a perfect match. The next band, if you look at, is present there. It's present in there, it's present there, nothing there. Again, a positive match on 2 occasions. If we go on to these two, and if we look at them together, you can clearly see that that has been similar to Jenny. If we go on to the next 2, we can clearly see that it's only found in that individual, as well as the last one. So guys, it's quite a simple process. It's looking for exact matching banding patterns. This wasn't particularly difficult for us to be able to identify. And the best way for us to be able to do that is to run a ruler down and tick off, and at the end you'll generate a band pattern that has exactly a match of that. So in this case, we've identified the individual, and the individual is Jenny, and you can clearly see how that banding pattern is a 100% match of that individual sample that was found. Okay, explain your answer to question 2, and that's important. For 2 marks, you must be able to explain the rationale for why you came to the conclusion that it was that specific individual. So you've got to explain yourself correctly and appropriately using the correct terms. So when we look at Jenny's DNA profile bands, there's a match or a perfect match with the DNA profile bands taken from the crime scene. If you look at it, it means that because there's a perfect match, it means that these 2 samples have to have come from the same individual. And in this case, we know that the sample of DNA that was found is a match to Jenny. And so that question is, now we've got to identify what was Jenny's reason for being there, or the reason for the blood having been on the window pane. And those are, again, information that we need to further investigate at a crime scene. State the next question. State one other use of this technique identified in question 1. So the technique is DNA profiling. As I mentioned, it's used firstly in solving crimes. And that's the principle based on identifying individuals and finding matching DNA samples to that individual. That's kind of the most convenient and easiest answer that we can use. But apart from that, we know that DNA fingerprinting can be used to prove paternity. Now guys, paternity again means relatedness to a set of parents. We often see questions in an exam where it references babies that were mixed up at the time of birth, and so we need to then use the DNA of 4 possible parents, set of parents, to link a child to each of those parents, and that's working out paternity, so the parents. We also know that individuals that go missing and often can be established. So there's lots of human trafficking around the world that happens, and so it can link, DNA sampling can link individuals to a set of parents as well as to an individual that has DNA being stored in a DNA data bank. The identification of genetic disorders. So another technique where DNA fingerprinting works is the ability to be able to screen individuals and look for genetic disorders in their genetic makeup. And that's a fantastic discovery that has led to becoming more clinical in the treatment that we administer to people. Establishing family relations, so again, in terms of siblings as well as individuals related to parents, we know that siblings will have some percentage of match of DNA. In relation to the parents, you'll find that that complement of DNA that you and I have is made up of genetic information from both parents. So we can establish relatedness by looking at DNA fingerprinting. Matching tissues for organ transplants. Guys, one of the challenges of organ transplants is that there is often what we refer to as tissue incompatibility. And that means the tissues often from different individuals are not compatible. One of the screening methods in terms of looking at compatibility between a donor and a recipient is to do a genetic fingerprint. And that again looks at typing the genetic information to see how similar their genetic profiles could be. And that often reduces the risk of rejection of organs during transplants. And then lastly, identifying dead persons, animals, etc. So as I mentioned earlier on, when a suspect has been found with, for example, a rhino horn, in South Africa, we're able to there's a project that has fingerprinted all rhinos around in the country. And so that allows an individual that has or is found in possession with the rhino horn to be able to track us to a specific rhino and where that rhino is from. So that can then link an individual to a crime scene. So it's important that the law now has evidence to be able to link individuals to a specific crime scene. It's also done for the trade of some of the exotic plant species like cycads, which are often traded on an international market. And so this can be used in terms of genetic fingerprinting plants as well. So that's an interesting question. As I mentioned, it's important that we recognize that there are essays in paper 1 and paper 2 that often can include sections on DNA, RNA, and protein synthesis. So let's look at one specific question and see how we would answer that question. So the question reads, describe the location and structure of DNA, the process of DNA replication, as well as the significance of the process of mitosis. So guys, as we unpack the question, it's important that we recognize what are the different parts of this question. And this helps in planning. And I often remind you that in writing an essay, it's important that we read the question well. We identify the different segments or parts of the question and that we brainstorm in a plan before we get down to writing the essay. This all requires time, but if done well, it ensures that you're able to comprehensively answer your question when you have to pen down your ideas for that question. So let's look at how I would go about doing that. Remember that the content is 17 marks. We have a 3-mark allocation for the synthesis, and I'll reference what synthesis is later on when we look at the answers. And it's key to understand that there are no marks awarded for answers in the form of tables, flowcharts, or diagrams. So if you decided that you learnt the process of, in this case, DNA replication, it will be good if you use that diagram to help you explain your answer. Unfortunately, you will not be given the mark for having a diagram that illustrates the process of replication. So yes, it's important to use diagrams to understand the process, but in the essay, it's important that we look at how content is synthesized into, in this case, the process of DNA replication. So, as I've illustrated on the next slide, I've had to take the question and unpack that into the 3 different components. In this case, the first part of the essay describes the process of DNA replication. The other part needs us to describe the location and structure of DNA. So we're going to spend some time allocated to the location and structure of DNA in our essay. And finally, describe the significance of the process of mitosis. So guys, it's important that we recognize that in any of your papers that several concepts can be linked together in an essay. And that's essentially how we're able to synthesize and string together, in this case, a response to the question. So in your answer, you've got to start off with describing the location and the structure of DNA. So where DNA is found, describe what are the building blocks of DNA, double helical, the nucleotides, the nitrogenous bases, and the complementary base pairs. I'll go through a suggested answer for that. We also then need to look at the process of DNA replication. Again, if I were to break up the 17 marks, I would simply allocate 7 marks to this, probably 5 to that, and then another 5 to that. That's just my understanding before even attempting the question. So if I'm able to get 5 significantly important facts for the description of the location of the structure of the DNA, and then another 5 for describing the importance of the process of mitosis, I would have had a kind of a clear overview now of what the mark allocation would be, so that when I write them, I note that at least 5 points and another 5 there, and possibly 7 for the process of replication will give me a total of 17. Okay, so in terms of describing the location, it's important that we recognize that part of the question required us to describe the location. We know that DNA is found in the nucleus as well as the mitochondria, and make special emphasis of the chloroplast in plant cells as an organelle that contains DNA. Then in terms of discussing the structure of DNA, we've got to be able to describe any of these 7 facts. So DNA is double-stranded. It basically is a helical molecule. It's made up of monomers called nucleotides. Each nucleotide consists of 3 parts: a sugar, a phosphate, and a nitrogenous bases. And then systematically discuss the 4 different types of nitrogenous bases. And finally, include the concept of complementary base pairing and how they are held together with hydrogen bonds. So if you think about it, it's quite systematic in terms of the way you can arrange your answer, and think about it logically. Think of the diagram, double-stranded helical structure. Then you discuss the nucleotides, and you also mention the base pairs and how they're bonded together. Okay. Also part of the question required us to be able to explain the process of DNA replication. And if you look at the mark allocation to this, was 6 marks. And so again, if you are traditionally inclined to being able to have that image of DNA replication in your head, it makes it easier for you to be able to string together this process. Again, guys, remember that this is only my suggested memo. We're expecting you to be able to write in a congruent flow of ideas and concepts. So please do not just bullet point them, because that's not what is expected in the essay. It needs to be ideas and concepts that are well strung together. So the DNA, you've got to describe it unwinding. You've got to talk about unzipping and the hydrogen bonds breaking. This then causes the strands to separate out, forming 2 templates. These templates then serve as strands on which the free nucleotides or the complementary DNA nucleotides will bond, where adenine to thymine and guanine to cytosine base pair. And this is using the free nucleotides in the nucleoplasm. This results eventually at the end in the production of 2 new identical DNA molecules, each molecule consisting of 1 original strand and 1 new strand. And that's essentially the overview of the process of DNA replication. Remember that the third part of the question was discuss the significance of DNA replication for mitosis. And guys, we know that mitosis was a process in which the cell copies and produces 2 identical cells. And we know that during interphase, we know that DNA replication occurs. So in this bit, you've got to be able to quickly describe that genetic material is double-stranded and that each cell receives the same amount of DNA, and it's important that the DNA replication takes place to ensure that the daughter cells have a genetically identical copy of that information. And finally, the synthesis of this essay is based on the 3 criteria: looking at the relevance of your facts, looking at the logical sequence of how concepts are linked together, and how comprehensive you are in terms of putting together your information. So often learners tend to forget about these 3 important marks, and that's crucially important in terms of knowing that there's no irrelevant information, that your information is logical and sequential, that you're describing processes like protein synthesis or DNA replication correctly and logically, and then finally being able to, in this case, the significance of a process such as mitosis. So guys, that's a wrap for this lesson. We've looked at an overview of all the concepts around DNA and the code of life. We've looked at various different questions on how we apply our understanding. I trust that you've enjoyed the lesson. Please go and attempt some questions. Take out those textbooks, look at the revision activities in them, download some papers, attempt these questions, and I hope that you improve your understanding. From my side, cheers, have a bye lekker day.